everyone. So now we are going to start with today's keynote speaker, Dr. John Smol. So Dr. John is a distinguished university professor in the Department of Biology at Queen's University, where he also held the Canada Research Chair in Environmental Change. He founded and co-directs the Paleoecological Environmental Assessment and Research Lab, which is a group of about 40 students and other scientists that are dedicated to the study of long-term global environment change, and especially as it relates to lake ecosystems. So much of his research deals with the impacts of climate change, acidification, eutrophication, contaminant transport, and other environmental stressors. John Smol was the founding editor of the Journal of Paleolimnology and is current editor of Environmental Reviews. He has received many awards, and more recently, he was elected president of the Academy of Science, Royal Society of Canada. So today, he's gonna talk about studying the long-term effects of climate change in a multiple stressor world. It is as clear as mud. So when you're ready, Dr. John, please go ahead.
Well, thank you very much. And thank you for the invitation. I'm very happy to be here. And I think this is a remarkably good initiative of our two joint societies. And I hope this continues. I think it's really a great idea. And especially now, since we have the technology with Zoom. I was asked to give this keynote some months ago, and I wrote down some notes when I was asked to do it. What I was asked was a combination of a keynote on how we can study the effects of climate change, and also to some extent advice. So I have a few pieces of advice here and there interspersed to early career researchers along the way. A key part of my talk, though, is it's on the effects of climate change. I'm not really going to talk about how paleolimnology has been used quite successfully to track climate change. I think that is well established. I'm going to look at some of the effects of climate change and how we can use paleolimnology to try and sort some of those issues out. And again, I'm giving this talk, but a large amount of the data was collected by many graduate students and postdoctoral fellows at my lab here at Pearl at Queen's University. And of course, I'm talking to an audience that is well versed in this, but these are some of the typical we live in a very complex world. A very complex environmental world. And to answer many of these questions that we deal with environmental policy, we typically ask these types of questions. We want to know if we think humans are causing a problem, we want to know what was it like before humans were part of the ecosystem or a large part of the ecosystem. We want to know what were pre-disturbance conditions. We want to know what is the range of natural variability. And we want to know have conditions changed, how much, how fast, when, and why. And each of these questions has a time component associated with it. And as we all know, the past matters. And the past matters because we have very little monitoring data. I did this analysis, well, I guess now it's almost 3 years ago. It's published in the Proceedings of the Royal Society of London B series. And I asked the simple question, how long are most monitoring programs? I went to the International Journal Environmental Monitoring and Assessment. I went to the previous year. And I asked, how long are most aquatic studies that are published in aquatic monitoring and assessment? We looked over 400 papers. And as you can see, about 2/3 are less than 1 year in duration, and over 90% are less than 3 years in duration. Now, that's not to say 3 years or less is not enough time to answer important questions, but it's hard to answer those questions that we posed in the previous slide. So we try and push that record back in time, and we do that in different ways. We do that whatever historical records we have, and I'm going to use all these methods really in my talk here today, try and use historical records, it could be meteorological data, it could be pushing the record back in time, looking at diaries or different types of records, but usually these get worse and worse as you go far back in time. Especially in the Canadian context, but also in other countries that are represented by the audience, we have what we refer to as traditional knowledge. This is, and in my case, it'll be primarily in the Arctic or the subarctic, where northern peoples have lived for thousands of years. They have not written down these records, but they have a strong oral tradition. They talk to their grandparents and their great-grandparents, and they know a lot about the land they live in, which of course incorporates a lot of environmental information. There's modeling. I'm not really going to be talking too much about modeling here, but we work with modelers. That's a way of also trying to forecast the future. And of course, we're talking about natural archives here, and especially lake sediments. And I'm going to be talking here primarily about the last 200 years. In Canada, where I live, that really is the main record of human impacts, of major human impacts. And for that, we need good surface sediment cores. Here's my colleague, Brian Cumming, using one of the glue cores that were developed in my lab for the acid rain work. But these are the high resolution sediment cores. We section them at typically close intervals. And then we look at a whole spectrum of proxy data. I'm primarily a biologist, but we also do some geological and physical proxies. But we can track, for example, what has happened what has happened from contaminants, from PCB, DDT, so on and so forth. We work on pollen, as many of you do, reconstructing terrestrial vegetation. And of course, we're primarily limnologists, and we're trying to reconstruct what happened in the lake itself. And foremost among these would probably be diatoms and other algal fossils, but also things like chironomids, Cladostria, and a whole spectrum of other issues. And we have to realize, we have to grasp the idea, we live in a very complicated world, the so-called Anthropocene, which hasn't quite been defined yet, but we're dealing with very, very different types of stressors. And so in this multiple stressor world, it's very difficult to tease apart what is the effect of aerial deposition, of DDT, of nutrients. And on top of that, we have a warming planet. And on top of that, if we're doing paleolimnology, we have the complexities of what actually makes it into the sediment record. So I think we have to embrace these problems. Having said that, I think using well-designed studies and often using a comparative approach of understanding the limnology of lakes, and that's going to be key. You have to understand the limnology of lakes, and then using that information to try and reconstruct the past. So when it comes to the advice part, I have a few pieces of advice here, just at the very beginning, and just what Fernanda had asked me to do, so I just do that at the very start. I asked the first piece of advice on that. Sometimes I think we're forgetting the limnology in paleolimnology. Or stated more plainly, we have access to powerful approaches. We've already saw many of them here this morning, yesterday, and when using paleolimnology, but only if we understand the fundamentals. Paleolimnology is based on those fundamentals. So if I build a castle of the foundations of paleolimnology, it's really based on the basic sciences of physics, chemistry, biology, which all of limnology is based on. So you first have to primarily be a limnologist, and you have to have at least a working knowledge background of physics, chemistry, and biology to be a reputable limnologist. Then we even push it further, and I'll just talk about biology. I'm primarily a biologist. I work mainly on diatoms and Cladostria. There, we need a fundamental understanding of ecology and also physiology. If you're going to use biological proxy data, it has to be connected to what the algal, if you were say diatoms, there's a large body of work on algal diatom physiology. You have to have a background in that to understand it properly, or else you can be easily fooled by simple patterns in lake sediments. And you have to understand the ecology. And that, again, is intimately associated with this limnological world. Then on top of that, where we're even pushing it a little further, we have sedimentary processes, other filters, and on top of that, I like to think paleolimnology is the king of all the sciences here, but it is based on this foundation. And so we have to keep that in mind. And don't forget the physics, chemistry, and biology when we try and do our interpretations. How do we do that? Well, it's very important to read journals like Journal of Paleolimnology, Quaternary Research, Quaternary Science News, the Holocene, but not at the expense of, for example, limnology and oceanography, plant physiology, animal physiology, journal of phycology. We need to keep that foundation. We have to split our reading, and it's hard. I mean, it's hard enough just to keep up, or it's impossible to keep up actually with this first part, but we have to keep up with the second part as well. ECR advice number 2, I think with a strong foundation in limnological principles, the comparative paleolimnological approach is powerful. Now, you need that foundation of limnology to understand how lakes function before you start comparing them on time scales. And what's fundamental here is that certainly not all lakes will respond similarly to the same forcing. And I think we're getting better at this, but for a long time, you'd say, well, the lakes, I have 5 cores and the cores changed at different times and climate changed at the same time, so it can't be climate. I mean, that just showed a fundamental disconnect from limnology. I mean, I work a lot in the high Arctic, for example. Here I show 4 sets of lakes, maybe 5 sets if you look in the background, 4 sets of lakes I'd see in one hour helicopter run if I was doing a helicopter survey of lakes in the high Arctic. We should never expect these different types, and then the background is Lake Hazen, still ice covered. This lake is gonna, if you take a core of this lake, you should expect it to have a similar pattern to all the other lakes in this area. If you understand the limnology, you can use that information to better understand what's actually happened, but you need that foundation in limnology. And finally, when using biological proxies, not all species changes are equally important. And this is why it's critical to have that understanding of the ecology and the physiology of the organisms. There are certain organisms, certain taxa that are very closely related. And you might switch from one to the other, and it might look like a massive change in a profile, but ecologically or physiologically, it's not that much of a change if you're thinking of the whole lake ecosystem. Similarly, you can have like sometimes 5, I give an Arctic example here. I do a lot of work in the Arctic. There are these small cushion plants in the Arctic. I could go to one Arctic ecosystem and see 6 small cushion plants. Then if I went to another Arctic ecosystem and I saw 2 cushion plants and 2 trees, well, the 6 on species diversity, for example, the place where I saw 6 cushion plants has higher species diversity than the 4 species in the other place. But certainly if I have 2 cushion plants and 2 trees, that's ecologically a much higher diversity. So again, not all species changes are equal. So we have to have that foundational work. Okay, so now with that advice, I'm going to move to 2 examples. My first example is looking at flood frequency. And this example is because Fernanda, who's one of the main organizers of these meetings, asked me to use this example. She saw me give it in a keynote in Mexico, I think some years ago. So some of these data are a little older. They're not like what we just published last year, but it does show how we can use, and again, I'm talking about effects of climate change. I'm working on areas where we know what the climate has. So we want to look at the effects of climate change, not reconstructing climate change here. And I asked the question, can paleolimnology be used to undertake risk assessment of ocean flooding? And we all see about ocean flooding. We tend to think of hurricane flooding with, episodic climate events. We tend to think of it, at least I do, tend to give it, it's happening in Florida, or it's happening in the Caribbean, or it's happening in more southern regions. And in fact, that is where the major flood frequencies are happening. But I'm going to take you to a part of Canada where you might not think this happens, and it hasn't happened commonly. But I'm going to take you up to the Canadian Arctic, where we looked at a major storm surge event that happened in 1999. And I'll put down, this was published in Proceedings of the National Academy of Sciences some years ago. But we did more work afterwards. And what I want to use here is to show how we can blend different approaches, different time scales, different lakes, and to come up what we think is a coherent picture. So I'm going to take you to the Canadian subarctic. Now, the examples I just showed you of those lakes in the Arctic, they're from up here, way up there. But I'm going to take it. Well, so I live down here in Kingston, Ontario, right on the edge of Lake Ontario. It's about 2 hours south of Ottawa, where you may know. I'm going to take you, for me, this isn't high Arctic. For some people it is, because I work usually up here. But we've done work throughout the circumpolar Arctic regions. I'm going to take you to the Mackenzie Delta. It's the delta of the Mackenzie River, one of the major rivers of the world. And what happened in 1999? So it's in the recent past. We have a history of this. What happened in 1999 is it started off by, in 24th of September, winds started blowing, strong winds started blowing, and they started blowing. And then with those strong winds, pushed water, and it pushed water inland. It's quite remarkable. The storm lasted 48 hours. Because this is such a flat area, this is quite remarkable. It pushed inland 20 to 30 kilometers, and it flooded this area with over 2 meters of seawater, freezing seawater. If anyone was there, they'd be dead. You couldn't last 48 hours in over 2 meters of freezing seawater. If any animal was on the ground or couldn't fly away, it'd be dead. But this is what happened. It happened in 1999, and it left a remarkable record. So just look back, it's a very flat area. It's an area also with lots of lakes, and it's a very low elevation. So this ocean water came because of this storm, and it moved in about 30 kilometers. It's quite remarkable. Okay, what was the effect of that? Well, this is what the subarctic tundra looks like. This is what it looks like typically in the unimpacted zone. If you went to the part that was flooded for 48 hours, it's now called the dead zone. It's called the dead zone because it pretty well killed everything. The salt water killed everything. So this is I can show you even this next diagram might be even clearer. The Arctic Ocean's in the background, that's 20 kilometers away. And you can see this line. This was inundated by the ocean. This didn't get ocean water. This is all dead. This is still green. These lakes are salty. You're probably getting what I'm getting at. These lakes are fresh. You can actually draw the line how far that ocean water got. In fact, you can see it in satellite images, and it's quite clear what happened. So we asked the question, we thought this was quite strange. We don't expect sort of this hurricane type inland movement of seawater in the Arctic. It sounds like something happens farther south. We asked, well, of course, there's no record. There's no written records of this. We asked, how common are these storm surges? We asked, are they really unprecedented? Because people said, well, this is unprecedented. But, well, how do we know? Maybe it happens could happen every 10, 20, 100 years. If no one's there writing it down, how would we know? So we used 3 approaches. And I want to just show how we tried to pull this all together. We used the oral history of the indigenous peoples. Now, no one actually lived directly right, but it was in the hunting grounds of local indigenous people where they go hunting. And so these people have been there for decades. And there's an oral tradition. The parents talk to their grandparents and great-grandparents. And so part of the study was to try and get an oral history of the area. Dendrochronology, I'm sure many of you are familiar with, tree ring analysis, and at the end, we brought in paleolimnology. So first of all, oral history. This was led by Steve Coquel, my colleague. We actually published this. We published it with some of the indigenous peoples. And we interviewed hunters and trappers who were from the area. And they said it has never happened before in their oral history. This massive type of storm surge has never happened. So that was the first piece of the puzzle. Okay, well, that's important. But that, human memory goes back maybe 100 years, but that's very important to know. Second thing we thought, what about tree rings? There are not trees per se, but there's shrubs, alders. You can see the dead alders here. Here are the living alders, here are the dead alders. My colleague Mike Pisaric went and did tree ring analysis, both in the dead zone and then the transition. And he did the tree ring analysis from these strings. And as you can see, here is a typical tree ring analysis. Before, in the 1990s, you see how wide the tree rings are? This is 1999. So this is not in the dead zone, but it's near where there was some salt water coming in. You can see right after some salt inundation, you can see how the tree was stressed and how the tree rings get smaller and smaller. The next thing was to find out, we have a lot of dead alders. When did they die? So you can tell that with tree rings as well. Sure enough, there was a mass mortality of shrubs in 1999 and continued on afterwards in salt. So the oral tradition says the 1999 is unprecedented. The tree rings say it's unprecedented. Now, how far? Tree rings, alders? Well, maybe 80 years. So we kind of got a story that at least in the last, say, 100 years, it's unprecedented. But maybe it happened 200 years ago. Maybe it happened 300 years ago. So that's where the paleolimnology comes in. So we looked at the maps and we decided to go core lakes that were in the dead zone and core lakes that are outside the dead zone. And we reasoned that since diatoms and other indicators are very closely linked to salinity, we should see a change in diatoms and other indicators every time the lakes were inundated by salt. So here's the first core we looked at. It's lead-210 dated, goes back about 300 years. This is a simplified diatom diagram. Red is salt diatoms, green is freshwater diatoms. I think this is the lead-210 date for 1999, the red line. I think as you can see quite clearly, there's a massive change in diatoms shifting from freshwater to marine type diatoms, brackish water diatoms at the time of the storm surge. And it never happened before. Yes, there's a few salt diatoms here and there. We're very close to the ocean. We get sea spray. That's not surprising, but there's never been such a massive increase over the last 300 years. Well, being paleolimnologists, we look at 300 years. We want to look farther. The next step is we take a piston core and we take it right to the bottom. These lakes aren't that old. They're only about 1,300, 1,400 years old. Again, the same color scheme. So now we've gone down there. Red is marine, green is fresh. Once again, I think you see the only place we have any significant saltwater diatoms is post 1999. It appears to be unprecedented in the history of the area. Now, I'm sure your eyes are going, John, you're saying, yeah, John, okay, I believe that maybe, but wait a second, there's a lot of other things happening here. We're not saying nothing else has happened. There's some massive ecological changes that happened before. And I think we know what they were. They're all fresh water, but these are primarily river diatoms. With Murray Hay back in his master's almost 20 years ago, we did a lot of work on the Mackenzie Delta. We calibrated the diatoms to river and lake forms. And you can actually make a loadic index, the amount of river influence. And what actually happens is that period is actually, those are river diatoms. And what actually happens, we're right next to the Mackenzie River. And over time, the river has captured some of these lakes. So this was a period of time when it actually was attached to a river. And so we can actually understand that because we have enough information to do that. These are river diatoms. So I'm not saying the ecology hasn't changed, but certainly we didn't have marine intrusions. So far we're all biological. Maybe we can do non-biological. Can we see it in the non-biological record? We work with Jesse Vermeer, and we ask the question, can we actually see this, for example, in particle size and things like that? Sure enough, the storm surge is very clear. The 1990s story with the marine intrusion, it was an erosion of particles of a specific size. Those clearly show up. We can also show that down here, just where we have the river diatoms, sure enough, where we have the river diatoms, we also have a river signal in the record. So I think we can use the biological record and the physical record and get that. Well, that was one lake. We said, well, we can do this on a landscape scale. So we did it actually on 6 lakes. I'm only going to show you a big summary here. We looked at different, again, a comparative approach, different elevations, different amount of salinity. And sure enough, there's the dead zone. Sure enough, as you get closer, lower elevation lakes, they have more, these are the marine diatoms, and just showing you simplified diagrams. It's all post 1999. As you go higher elevation, you get less marine diatoms, lower. So I think this shows quite clearly. Take a core outside of the dead zone, no marine diatoms. There's 1999, no red diatoms. So I think you can actually do this on a comparative scale, knowing the limnology. You can predict what you should see, and then you go and find it. What about other trophic levels? I won't go into that in detail, but we looked at Cladocera, we looked at Chironomids, we looked at, these are all published separately. But sure enough, they all really change right around 1999. There's other changes that are happening with climate and stuff, but the salinity indicators are all happening. So you can do this in multi-trophic levels. So then you would ask, okay, so I think we convinced ourselves 1999 was a very strange year. Why? Why did this happen? Well, the usual suspects. We know with warming, rising sea levels, you should get more inundation, true. We know we get more intense weather, true. But what is probably critical for this area is later freezing, decreased ice cover. As you hear all the time, ocean ice cover is decreasing. And what happened? Well, if this same storm happened in 1970s, we'd get no storm surge, because at the end of September, there was still sea ice. So you could blow as hard as you want, but you're not going to make a wave if you have sea ice. Now, with declining sea ice, it's already free of ice in September. And what happens is now you've got liquid water and you've got a storm coming into it. And with declining sea ice, we should see this more and more. And of course, we don't tend to think of Canadian Arctic as being susceptible to ocean incursion, but we have to think about this. almost all the Arctic indigenous communities are coastal. So this is something that even a place like Canada has to start thinking about. So I'm not saying it's the only cause, climate change was the only cause, but we've sort of loaded the dice for the frequency of disasters. And we're even seeing that in the Canadian high Arctic and subarctic. Okay, I'm going to take from the Arctic and I'm going to come down to near where I live in the temperate regions. And this is a paper we submitted. And, there's no question this is complicated, lakes in a warming world. And we see a whole spectrum of issues that are happening with warming. One of the most obvious ones is loss of ice cover. This is critical. Losing ice, if you're in an area that has ice, now you start losing the ice cover, this is critical. Because that changes everything. It changes light regimes, changes mixing regimes, surface warming, it's reduced the mixing. So now you have stronger stratification, you have higher evaporation, water levels are changing, you have reduced cold water fish, and you have what I'm going to get at in my second example here is increase in blue-green algal blooms. So what's actually happening? Well, this is an old diagram, but changing ice cover is changing tremendous amount. This is the same lake under a cold, warmer, and much warmer situation. The duration of ice cover affects almost everything. It affects the length of time, the growth. Now, I'm not never saying that you don't have algae growing under the ice. You do, but they're much reduced, and especially in places where you have 3 meters of ice. That alters the mixing pattern and the growing season. We have what I've been referring to as now a longer summer. With less ice cover, you have a longer summer, more time for algal production, more time for thermal stratification, and thermal stratification that is much more intense and lasting much longer into the summer and actually into the fall in some regions. This is perfect territory for algal blooms. And so I'm going to ask the question, climate change is longer summer, but why is my lake turning green? And what I want to look at is algal and blue-green or cyanobacterial blooms. No one wants blue-green algal or cyanobacterial blooms in their lakes. What's happening? This is just for Ontario. I live in Ontario in Canada. Many of the lakes here are by any standard, are considered very low production clean. Yet, even though they're low phosphorus, low nitrogen, look at this. This is the increase in complaints of blue-green algal blooms going up. Each year, we're getting more and more complaints of blue-green algal blooms. What's happening? Many of these lakes are oligotrophic. What's happening? So before I begin with this second example, just to make clear, without question, you need a minimum amount of nutrients, phosphorus and nitrogen, to generate algal biomass. So there's no question what was done in the 70s, before my time, showing how especially phosphorus is the driver of algal populations in most regions. This was done very clearly. There's no question. There's no question about that. What I am saying, people are recording algal blooms, increased algal blooms, even in lakes with steady, or in the examples I'm going to show you sometimes, declining phosphorus and nitrogen. That is a bit of a surprise, and not a happy surprise. And we asked the question, and others are asking it now too, but we've been asking it for a while, can climate change, could the less ice cover, the longer summer, the more thermal stratification, could that be affecting algal blooms even without increasing nutrients? So we're talking about the longer summer and enhanced thermal stratification. So ice cover is an obvious one. You can see that. But it also, as I said, has important implications. I'm going to take you to one lake called Lake of the Woods. We've done a lot of work. We have lots of examples, but I think this is a good one. I show it because many people, at least in North America, know where it is. It's almost a Great Lake. So I live here in Kingston, Ontario. So this is the border between Minnesota, Ontario, and Manitoba. It's an international lake. Part of it's in the United States. A very complex, very big lake, has bays. We have many bays. The bays are almost like mini lakes. It's sort of the same water, but some stratify, some don't, and we know where that is. Some have higher phosphorus, lower phosphorus. And so we have, like within this area, we can do cores from different bays. And it looks like a success story. Back in the 1970s, people realized, wow, we have a real problem with phosphorus. With very effective measures, they greatly reduce phosphorus. Nitrogen is down, phosphorus is down. Really, it looks like a success story. And it was for a while. And yet, Now, algal blooms are being reported. This is some days, they're closing out. This is in 2015. Algal blooms, especially blue-green algal blooms, are being reported in some bays. So what's happening? People are complaining about blue-green algal blooms. They're saying they're increased intensity, but what's going on? The phosphorus has gone down. So we thought we'd explore this in various bays in different areas. I'm just going to show, we have many examples. I'm just showing you some. We can use diatoms. Diatoms we can use to reconstruct phosphorus, and we've done that throughout the bays. Also, diatoms are very closely linked to the amount of ice cover and the amount of thermal stratification. That's been so well established now, I don't think I have to defend that. And so let's just take a look at one bay. Look at one bay. This is a bay. This is just part of the diatom diagram, obviously. I'm doing this for a presentation. This is just one bay, Whitefish Bay. And that's 1980, the Letu-10 1980. And look at the big change in these two species. Olacosyra, as we know, requires lots of mixing. It's a big, heavy diatom. We also know Discostella, these various small cyclotella, cyclotoid species, they are slow sinkers. They like the longer summer. That's well established. They also thrive typically with stratification. So basically you don't need the stratification, you just need the longer summer. But if you have a longer summer and stratification, they tend to be even higher. And of course, context is key, as Kathleen Rulin, Andrew Patterson, and I keep saying. You have to know the limnological context. You can't just say that. But if you understand the limnological context, you can say that much more strongly. Well, this is the typical kind of change we see with a regime shift in the lake figure. This is a well-mixed, to us, this is well-mixed diatom assemblages. This is a diatom assemblage that is now longer summer, more thermally stratified. And we can actually look at that. We actually have temperature data going at this area. There's actually a weather station, goes back to the 1900s. And in green is the temperature data. And in red is our paleocyclotella data. And you can see that's a pretty close relationship between the cyclotella and the temperature. You can see it's equally the drop in Olacosyra. This bay, they actually have ice records. They have ice records going back 50 years. And I think even this is even much stronger. As you can see, as the ice is declining, you can see very nicely the cyclotella, the fossil cyclotella is matching the known records of decline. Look at this. That's the Olacosyra, the one that likes the colder, well-mixed water. You can see how it's closely related to the ice out record. So we say these lakes, some of these bays are changing dramatically in their structure, in their thermal structure. Well, so that was established. Next step is we want to know, have they increased in algal blooms? How do we do that? Well, chlorophyll A is the main pigment in all algae, all plants actually, algae, cyanobacteria. So if we can track chlorophyll A, we'd have a sort of an estimate. And that's how we actually estimate even eutrophic, mesotrophic lakes. It's often measured on the chlorophyll A spectrum. If we can estimate that, there's different ways of doing that. One way is HPLC with the diagenetic products. We've been using spectral analysis. These techniques are being developed quite quickly. The methodology we're using was developed by our colleagues and ourselves in our lab, Neil McLuty in my lab. The algorithms in 2010, we did a review on it in 2016, Journal of Paleontology. I can send that to you. But it's a fast, non-destructive approach. Once you have the machine, which is about $100,000, it's a fairly fast system. And we can use that. Of course, you have to always worry about diagenesis, but the technique we're using, this is chlorophyll A up here. It goes through a series of diagenetic steps. It loses its magnesium because pheophytin. It loses this long chain here to represent R. It becomes pheophoribide. The technique we are using measures all these measures its isomers and these three products: chlorophyll A, pheophytin A, and pheophoribide. So our technique actually incorporates most of the diagenetic products. So as such, most of them are accounted for. Now, that's not to say it's 100%, of course. It never is. A recent paper by Rydberg et al., we weren't authors on this, but it shows very nicely that this technique is strong. They show strong confirmation, but there is some diagenesis, but usually in about the last 10 years or so, and it's minor. It's relatively minor compared to the magnitude of our changes. So just while I'm talking about chlorophyll, I just tell people we're using that approach also to reconstruct dissolved organic carbon, total carbon. That's Karsten Meyer-Jacob, a postdoc in my lab, has published on this. And we've also, in an exploratory way, working on using the same technique to look at cyanobacterial pigments, but this is still somewhat exploratory. But just to let you know these spectral techniques. But let's go back to chlorophyll A. I want to talk about chlorophyll A because that's algal production. And here I show you 3 bays of Lake of the Woods. And these are still very low phosphorus levels, 14, 12, and 10 is less than 10 is very low. Whitefish Bay is oligotrophic, and it warmed, but still no increase in chlorophyll A, no algal blooms. It hasn't crossed that threshold. We're starting to say, we don't want to say too loudly, but it sounds like around above 10 is where things start happening. These are still very low phosphorus levels, but look at this. Look at in Echo Bay or in Cul-de-sac Bay, we have increases in chlorophyll A, especially in the last 20 years, exactly during that period of intense warming. What do we think was happening? We've done this in many bays, but we've been going from bays that were largely well mixed with the old Lancashire and so forth. And with this accelerated warming, we have shifted with less ice cover, longer growing season, more thermal stratification. Thermal stratification, we know that's going farther and farther into September, sometimes into October. We've shifted to this kind of regime shift, weakly mixed, increased thermal stat. We have those discostellas, that cyclotella diatoms coming in. And of course, if anyone who works on blue-green algae and green algae in general, they like it hot. Even without further increases in nutrients, those areas that are up this threshold about 10 are starting to increase in algal blooms, just as the time when the climate warming and the thermal stratification was coming in. And on top of that, it's not just blue-green algae. It's not just algae, sorry. It's blue-green algae is what people are really worried. They form toxins and all these. That's the ones you really don't want. They especially like it hot. They especially like very well thermally stratified conditions. And they especially like that late summer when the nutrients are lower already and they can, using their vesicles, they fill with the gas vesicles, they come to the surface. It's perfect. It's like a perfect storm with this longer summer. So far, all I showed you is the total algal biome, just chlorophyll A. This is work that was done. We had nothing to do with this paper. It was published after ours, Pilon et al. In Lake and Reservoir Management. What they did is they looked at eDNA in Lake of the same bays, the same areas we were working on diatoms. And they also looked at microsystems, the actual toxins of these cyanobacteria preserved in sediments. And as you can see, these are dates down. This has nothing to do with us. We didn't do this, but the eDNA, this is all bacterial DNA. This is a proxy for blue-green algal abundance. There's another proxy for blue-green algae using the RNA in this case. And here is the microcystin synthesizing gene. As you can see, they're all increasing post-1980s. And then what they did is they took our cyclotella, they asked for our data, our diatom data. And if you put where we said the lake started thermally stratifying and so forth, and that that bay started losing its ice cover and thermally stratifying, so they superimposed our diatom data, and sure enough, where we had that regime shift is not only where we get more algal blooms, we know from chlorophyll A, but we're also getting blue-green algal blooms based on their data. So is there a disconnect? Well, the perception is correct, but warming is likely playing a key factor. This is not good news. It's very hard to decrease these algal blooms. It's very hard to get even lower phosphorus levels. So this is a challenge. I just have a few more slides to show an extreme case. Lake of the Woods, they're cottages. You know, there's still people around there. I'm going to take you to a place that you have to hike several kilometers to get to in Canada. This is this is Dixon Lake. It's a wilderness lake in Algonquin Provincial Park. This is a provincial park north of Toronto in Canada. This is a lot of Canada looks like this. Here, this is coming in by an airplane because it's not an easy place to get to. Look at this. It's a complete wilderness lake and there's this blue-green algal bloom happening. What is going on? So I'm going to take you to Dixon Lake. It's, as you can see, over 5-kilometer hikes. This is how you get there, carrying all your equipment. Certainly no watershed disturbance. But the phosphorus, look at the phosphorus. It's at that 10, 12, 13 level. It's totally isolated. Definitely no cottages, no nothing. And yet they started closing the lake to visitors. The hikers, they were closing it to hikers because they had a blue-green algal blooms. They had blue-green algal blooms to the point with toxins that they were worried they didn't want hikers going there. And it was caused by what used to be called anabina, electrospermum. They change the names all the time, so a little hard to track. So we asked the question, well, I mean, again, this is a wilderness area. Maybe you get these algal blooms every 10 years, 20 years. No one was sensitized to it. No one was there, really correlating algal blooms to anything. So we asked the question, what do we core this lake? Can we find out, are these algal blooms just now, and are they linked to climate? So this is the work with Liz Fafad, was part of her PhD. Again, we have the full diatoms, we have chironomids, we have we have the whole spectrum. But I just want to show you, this is the chlorophyll A profile for it. And as you can see, chlorophyll A, this is the line of previous maximum chlorophyll A. Chlorophyll A has changed a bit. We see this in a lot of Algonquin Park lakes. There's a whole other story here. There were declines in chlorophyll A for many years. That could be related to acid rain, could also be related to other issues, but that's not the story I want to tell you here. What we do see is, sure enough, since 2005, when people started recording algal blooms in this lake, there goes our chlorophyll A. But this is a blue-green algal bloom. These were these old Anavinia, they have aconites, as many of you know. These are the resting stages that are specific to this group. Not just, well, several other types of blue-green algae have aconites, but you can identify where they're from. Liz looked in the fossil record for these aconites. She said, when did this blue-green algal taxon start blooming? And sure enough, they're only there in any abundance in the surface sediments. While we have overall chlorophyll A, and yes, it's the cyanobacteria, and it's unprecedented, in this case, at least the last 300 years. Unprecedented levels of primary production and cyanobacterial activity. So this is happening, and we have many examples of this now happening recently, and is complicating the issue. I don't have, this is my last data slide. I'm just saying, initially I thought I had an hour, so I had the chironomid story here to say how deep water oxygen is changing, but there are many repercussions of this besides algal blooms, one of which is once those algae die, they decompose in the deep water and they start decreasing deep water oxygen. And so you've got things like trout, these prime species, these iconic species. Well, with climate warming, the surface waters are getting too hot, and the deep waters used to be a refuge, but now with these blooms that are decomposing, they don't have enough oxygen. So we get what we call an oxygen squeeze and a thermal squeeze. And so we're excluding a lot of these prize species like trout because it's too hot in the surface waters and there's not enough oxygen in the deep waters. And we can track that with chironomids, but that's another story. So, in Canada, big long winters, people say longer summers. They say, John, what are you talking about? That's great. Well, yeah, that's great to some extent, but you might not want to go swimming in this lake if that's actually what happens. So my conclusions is, with decreased size and snow cover, we have a longer summer. And I mean, there's no question that allows for more primary production. But in addition, because of these warmer summers, thermal stratification more intense, lasts longer. This is especially the perfect storm for cyanobacteria. And I didn't get into this because of time, but also wind speed is decreasing. Perfect storm for cyanobacteria, lack of mixing. And we're seeing algal blooms, especially cyanobacteria, with lower and lower phosphorus levels. Well, this has serious management implications. We already had a lot of fight to get to the phosphorus levels we're at. This is implying you have to get even lower, and that's not a simple story. And on top of that, this is a paper I'm a co-author on, but there's so many other threats out there happening simultaneously. These are these emerging threats, and climate change, I've been referred to it way back in 2010 as a threat multiplier. Virtually every problem I deal with in paleolimnology, in limnology, gets worse on a warming planet. And here's one for sure. So again, I think I'm speaking to the converted here, but the past matters. Only paleolimnology can often get at some of these answers. And I think with the new approaches that are coming, we're just going to get stronger and stronger in this field. So thank you very much. And I'm happy to take questions if there's time, or if not, you can email me later. Thank you very much.
Thank you, Dr. John, for this amazing presentation. Now we have time for some questions. The first question is in the chat from Johanna Clanton were photosynthetic pigments specified to cyanobacteria also shown to be useful to trace changes in thermal stratification?
Yes, yes, that's a very good question. The standard way to track cyanobacteria or blue-green algae is with specific pigments for them, using typically HPLC technology. And that is certainly I would call it the gold standard for doing pigment analysis. And I believe we might have a talk coming by Dr. Waters tomorrow or the day after. We'll probably be using that approach. That is a very powerful approach. It requires an HPLC, quite a bit of water chemistry and so forth, but it's a powerful approach. Some of these spectral approaches that we're doing, we think, and we have one paper already out on cyanobacteria, which we think, and we just had a second paper where we compare our spectral approach of cyanobacterial pigments with eDNA, and it matches quite nicely going back in time. We think some of these spectral approaches, because they're fast, they'd be much more accessible than with HPLC. But HPLC, the standard method, I would say is the gold standard. These are in some ways approximate and show trends. We're not even showing, I'm not even arguing concentrations so much, but they can show the trends in these different groups of organisms.
Sorry. Savas have a question too. She raised the hand. Yeah.
Can you hear me?
Yes.
Okay, perfect. I'm currently studying a PhD with Dr. Waters here at Auburn at the Polygovernmental Lab. My question is partially answered. Would you consider eDNA or spectral approaches a more powerful technique or pigments and HPLC? And is there a difference between the two regarding how long back in time we can go?
Sorry, what was the last part? You broke up a little bit.
Sorry. Is there a difference between the two techniques that you mentioned, the eDNA and the pigments, about how long back in time we can go?
With our how long back in time we can go? Is that what you're? Yeah. So with our spectral technique, we have one test where we did a very strange lake in Baffin Island. Usually in Canada, the lakes are 12,000 years old, last ice age. But we have these lakes on Baffin Island that were so covered by ice, the glacier, they didn't get scraped by the glacier. We have 3 interglacials, and we did our chlorophyll A technique. It's published in the Proceedings of the National Academy of Sciences. I can send you that. But we did three glacial periods, and we see the same types of patterns going back, in this case, over 200,000 years. So the first indication, it looks like it holds. And I know HPLC has been done in the past as well. eDNA is coming, I think, and I think there's other areas. I'm not the best one to speak about eDNA, but at least our spectral techniques seem to be working, going back about 200,000 years.
That's incredible. Thank you, and thank you for your presentation. It was very.
I can send you those papers if you email me. I'm happy to send them to you. But HPLC is, I want to say, the gold standard. If you're working on HPLC, good for you.
Yes, thank you.
So I saw that Karina raised her hand, but I have a question before on the chat by Itzel Sigala Regalado. Thank you for your talk. It is always very inspiring to see all that we can achieve through paleolimnological studies. And I would like to ask you what you think about the new research methodologies in lakes using satellite images to know about eutrophication.
Yeah, that's a very, you see my gray hair. The technology when I was your age was air photos taken every 30 years or something. We really have now quite a way of blending with satellite imagery, which is showing present day blooms and present day conditions, and they seem to be getting more and more powerful each time. But also we have a sufficient record in some of these areas that we can use those different images going back in time to depend which part of the world you're in, going back in time. And so, I mean, that is a way of sort of ground truthing what the satellite imagery is showing and what the paleo data are showing. And I think that satellite imagery is showing, and it's showing us vast amounts of spatial scales, and we can show the temporal scale. So there's some temporal scale in these satellite images, depending how far back they go. But I think working with these people, paleolimnologists working with these people, now we have everything. We have the spatial and the temporal. So I think we have a very important niche to play in these types of analyses.
So we have time for our last question. Karina, go ahead.
Hi, many thanks for your talk. It was really great. One of my question is regarding Akinet, and you answer a little bit about pigments, how long they can be preserved, and if you can recommend some technique to study soft body algae in paleo records.
Yeah, so aconites are very resistant. Of the soft algae, I think they're very good. They're done because, and people have regenerated them that are decades and decades old in this sort of resurrection ecology. There certainly Bos van Giel in the Netherlands has done a lot of work on these soft algae, and some of these aconites go back. I'm not sure how far back, but certainly full post-glacial people have found them the last 12,000 years. The other soft algae, I think there's been less understanding of how of preservation issues. We have a couple of people in our lab, Pearl, not my side, but Brian Cummings' side, who have looked at them. They really is quite interesting, and they seem to, if I look at them under a microscope, I see some of them 12,000 years ago. They look pretty good to me. So, but I guess we've done far less on the soft algae as we have like on diatoms or things like that. But if we can get at soft algae, I think I think it's important. And there, pediastrum's been used for a long time because pediastrum's found on pollen slides. So the palynologists have been finding pediastrum from Van Post's time, I think. But so I think all the proxy data you can use is certainly, I'd use it.
Yes, and I was thinking, another question, sorry, is I was thinking with your presentation that maybe in the recent time, we need more and more proxies like this soft algae or pigments together with diatom, for example, because if I was thinking that this kind of bloom usually happen in the near shore. And if we only are working with diatoms and we see an increase in, for example, epiphytic diatoms or benthic diatoms, we could think that maybe it's a shrink in the lake or something like that. And this can give us a different interpretation, right? So
yeah. The more you have, the stronger you can say. And also, if you can do Cladocera, even get different groups, people, animals, plants, algae, I think you have a much more powerful situation.
Thank you.
Thank you. I'm afraid we don't have any time left for more questions. I see we have another question in the chat, but you can about from Winging. So you can make the questions through the YouTube channel because we have to move on. I'm sorry.